Well, here we are. We have the great privilege of another hour and a half of some insight. And certainly, I think we're going to close out the day kind of tying things together. That is, Dr. McKnight is here to talk about regulatory proteins and translation. And really, both in sickness and in health, we are the products of our genes and ultimately what the proteins do in some ways as we interact with our environment. And Dr. McKnight has made great contributions both in the academic and commercial space, and he has lots of accolades. But one of the things I always do when I'm going to introduce someone is I just do a simple Google search, because I'm a human being. And number 5 on Dr. McKnight's Google search is a wiki in German. Can't find a wiki in English, but you can find a wiki in German. So I went to figure out why, and I think it said he sits on a board in Vienna. So we're thankful for your global leadership, and look forward to hearing what you can share.
I love brief introductions. Fantastic. This first sounds loud. Is that better? Can people hear me? Well, it's a delight to be here. And I met Larry probably 35 years ago right here in this department. And I took to him right away because he's irreverent. And I'm sure all of you know this. There was a kind of an unfriendly virologist at the time, whose name will not be mentioned, who I didn't like at all. And we were talking about this person, and Larry said, "Oh yeah, he's a pig." Irreverence. But also, he's done so much for your community, for this school, and for biotech in this area, and just a devoted person to science in every regard. And this celebration, and I guess this is the 7th, is a wonderful testament to what you've done. We really appreciate it. Okay, I'm going to talk about the fusion of 2 enigmas, long-standing enigmas in biology. The first of which goes back over 100 years, and it deals with puncta in cells that have no membrane around them. So the typical cell has a membrane around it, everyone knows that. And the typical organelles within a cell also have a membrane, the lysosome or the mitochondria. So we understand how cells work in part because they're compartmentalized and they have surrounding membranes. But for over 100 years, we've known about puncta in cells that don't have a surrounding membrane, and that goes all the way back to Ramon Cajal. And the first person who did an experiment relating to these non-membrane bound puncta was a zoologist by the name of Robert Hegner. And so what Hegner noticed is he could see in the eggs of beetles, at the posterior tip, he could see these little puncta, and he wondered what they were. He could see them very clearly. They were basophilic, so they must have had a nucleic acid and RNA in them. And he wondered what were they doing at the very tip of this beetle. And he knew they were there, he could see them, but to get at function, he did a really cool experiment. He took a hot needle and stuck it into the posterior tip of the beetle egg, and then let the egg grow up. And the egg grew up fine, and a beetle came out, but it was sterile. It couldn't have babies. And he inferred from that those little puncta at the posterior tip carried the information for the germ lineage to allow that organism to have gonads and reproduce. And he was exactly right. I mean, that's a really cool experiment done in 1908. But no one knew what these were. More recently, they've been studied by many people, including Susan Strom. And didn't Susan Strom work here at one point with Bill Wood? Susan did great work on these same sorts of puncta in C. elegans embryos, and Tony Mahowal at Indiana did beautiful work in fruit flies. And let me go to the next slide, first slide, and show you what these look like. So here is a C. elegans embryo, and it divides in 2, and it starts out with these puncta, then it divides in 2, and the puncta move to this cell, and eventually they do the same thing. They go to the very posterior tip, and they serve the same purpose as in the beetle. They specify the germ lineage. So here are these germ granules in C. elegans that you can see. You know that they're there, but they have no membrane around them. So the enigma here is, what is the organizational principle? What organizes these if they have no membrane around them? What puts them together? So that's the first enigma I want to talk about. And these are kind of all over biology. One of the coolest places is in nerve cells. So in a nerve cell, here's your cell body with the nucleus. That's where all the genes of the nerve cell are. And so the genes get transcribed there, and they make messenger RNAs. And most of the messenger RNAs are translated in that soma, in that germ cell, in that nerve cell. But certain messenger RNAs don't get translated there. They get put in these same granules, RNA granules, and they get transported down the nerve cell so that they can be translated at the right site of the cell. And the right site for many messenger RNAs is the synapse. And so those little granules allow the messenger RNAs not to be translated up here, but to be moved down the nerve cell for appropriate translation where they're needed. And this can be a meter. So it can go from your spinal cord to your toe. So you're going to have to move a messenger RNA a meter all the way down that dendritic process for localized translation, because that's just a much more sensible way of doing things rather than translating each of those proteins and moving the proteins one at a time down the nerve cell. So the first enigma that I want to talk about is these granules. What organizes these granules? What's the organizational principle? So the second enigma is these gibberish protein sequences. So we call these low complexity sequences, and I'll explain what that means in just a minute. But there are kind of two kinds of proteins. There's one kind of protein that knows how to fold and make a three-dimensional structure. Enzymes know how to do this, and we, over the years, in the context of my own field, transcription, gene expression, we're able to purify these proteins and resolve their structure and understand exactly how they work, how they could bind a gene, do the right thing. And by understanding the structure, we could understand function. And by slowly doing this over the years, we have pieced together much of this part of the puzzle. But all of these regulatory proteins, transcription factors and RNA binding proteins, not only had this well-behaved, properly folded domain, but they also had domains that when we would purify them, were not folded. They just floated in space. And they were very susceptible to proteolysis. They were a pain in the butt to work with. And so we just learned early on to get rid of those, clip them off and get rid of them. But that was foolish because we knew these domains were really important. If you didn't have that domain, the transcription factor couldn't turn genes on. It could bind to the genes with this domain, but it couldn't flip them on. So we knew this was really, really important, but we couldn't figure it out. And back then, 30 years ago, I found one called viral protein 16, BP16, and it's been used all over the world. And it's a fantastic activation domain, but I never figured it out. I just gave up on it. And so that's the second enigma. How do these work? So 20% of our proteome is this stuff. 80% is well-behaved and ordered. 20% is garbage. And we don't know how that works. So if you could figure out how that worked, you'd be offering a nice step forward. So let me show you one of these and show you why I call them low complexity. So this is the low complexity domain of a protein called fuse, fused in sarcoma. And I'll have more to say about that shortly. So here's the low complexity domain, the gibberish domain of this fuse protein. It's its first 211 amino acids. And let's look at this sequence and ask, what is it composed of? So here are the 20 amino acids, and any normal protein is going to use all the letters in the code to be able to fold and do what it needs to do. So what does this one do? 83% of the amino acids are only glycine, serine, glutamine, and tyrosine. So it's low complexity. It's not using the full deck. It has 0 phenylalanine, 0 tryptophan, 0 isoleucine, 0 leucine, 0 valines, 0 histidine, 0. So it's not playing with the full deck. It's low complexity. So in reality, it looks much more like RNA or DNA because it's only using 4 letters of its code. So it seems like a really foolish way to use a protein. We can understand why they don't fold. They don't have the appropriate code words to be able to fold in the appropriate way. So how could these possibly work? So about 4 years ago or 5 years ago, we stumbled onto this crazy observation. So let me start you up here with this protein, fused in sarcoma. It's an RNA binding protein that has well-ordered components that allow it to bind to RNA. So it has parts of the protein that fold up just right and behave perfectly. But it also has this low complexity domain, the first 200 amino acids. And this is a really important protein because it can be mutated in 2 different ways. Certain of the mutations of this protein cause ALS, neurodegenerative disease. So it's a really important protein in the context of neurodegenerative disease. But the way it got its name, fuse, is fused in sarcoma. And what happens in that mutation is this low complexity domain gets translocated, separated from the rest of this protein, and stuck onto a transcription factor DNA binding domain. And that's the causative event of many forms of childhood cancer. So you can mess with this protein and get cancer if you put this onto the transcription factor, or if you mutate it in any one of a number of places, it can cause amyotrophic lateral sclerosis or frontotemporal dementia. So it had a low complexity domain, and we're working with that domain, and we left the preparation in the test tube, and I came back 2 or 3 days later and opened the test tube, and this protein, this purified protein, it formed a gel. It formed like Jell-O in the tube. And so if we fused it to glutathione transferase, the gel would be clear. If we fused it to M-cherry, the gel would be reddish or pink. If we fused it to green fluorescent protein, the gel would be yellow. Well, in all my years of working in biochemistry, I'd never had a protein do this. I'd had proteins do lots of things, but this never happened. So I said, this has got to be cool. I'm kind of a fool, but when I see something new, I say, it's like a new toy. I said, this has got to be cool. So we thought maybe this could tell us something about how this domain worked. And so with Masato Kato and Tina Han and some wonderful trainees in the lab, we turned this gelation phenomenon into an assay. So what we could do is we could make a little teeny droplet, hydrogel droplet, where we fused M-cherry, red, onto this low complexity domain, and we would get a little droplet, a little gel. Now we could put that into a chamber slide, and we could challenge it with test proteins. We could do things with it, and that became our assay. And for a biochemist, you have to have an assay. Without an assay, you can't do anything. And so this became our assay. And so one of the first things that we did was just ask, what might this be able to do? And so we did the very simple experiment of taking that hydrogel droplet, exposing it to simply green fluorescent protein, and it was porous, and so the GFP got right into the gel, but it washed right out. So there's nothing that would attract the GFP into the hydrogel droplet. And yet if we made GFP with the same sequences, those same 214 residues that were used to make the gel, now that test protein would get trapped. So the gel could trap that test protein, and now we had an assay. So now what we could do is mutagenize this randomly, just mutagenize it like crazy, characterize what those mutations would do to binding to the gel droplet, and compare that with the ability of this fuse protein to go into these granules, into these non-membrane granules. And we could say, is there a correlation between the two? And the correlation was spectacular. So we could say, we don't know what's going on here, but whatever's going on here may tell us something about how these granules form. So that's the idea. So we've done that till the cows come home, and I'm not going to go over a bunch of that data. It's published, and it would just bore you. But we could do a lot of other things. So we could take these gel droplets, and just take the cell extract and put the cell extract on and then ask what could it bind? Well, it bound all the things that go into the granules. It bound all the RNAs. So it gave us an assay, and we could use that to begin to convince ourselves that something about this was telling us something about how cells work. So I have 2 slides to kind of summarize kind of our thinking on this. So here's a typical RNA granule protein, and it has folded domains that behave perfectly, that form a beautiful structure, and allow the protein to bind to the right messenger RNAs to take them into that granule. So that's one component. But it also has these low complexity sequences, and we think of these like Velcro to bring this domain and its attached RNA into the RNA granule. So we kind of have 2 parts to this. One, the conventional part of proteins that fold very nicely and do the right job of binding the right messenger RNAs, and then this Velcro to bring them into the RNA granule. So we could kind of concoct this back together in a very trivial way. And this is a Ganonkin experiment. It had to work, so don't be impressed. But it helps me to convey to you the way we're thinking about this. So we start with our gel droplet made of the low complexity domain of fuse. So that's our test. And then we take cyan fluorescent protein, which is kind of bluish. And if we take cyan fluorescent protein and put it onto this, it won't bind. Then we add on some bells and whistles. So for the RNA binding domain, we use the MS2 phage coat protein that folds up perfectly and knows how to bind this hairpin. And we put a fluorescent group on the hairpin so that we can trace the RNA as well as the protein. So if we put the MS2 RNA binding domain on and ask what happens, nothing happens. It doesn't work because it has no Velcro. Now onto the CFP, you add the Velcro, the low complexity sequence of fuse. Now the CFP can be bound, of course, because it's got the low complexity sequence that is the Velcro for binding, but it can't bind the RNA because it doesn't have the RNA binding domain. But if you satisfy both components, you have the Velcro and the RNA binding domain, now not only will the protein bind, but it'll bring the hairpin RNA into the hydrogel droplet. So this has been our mainstay assay. And it's allowed us to probe into this and understand it in considerable detail. But now we've got to deal with a real big problem. What in the world is this? It's a gel droplet, and I'd worked as a graduate student at the University of Virginia with Oscar Miller, who had worked with Joe Gall. And Oscar and Joe always profited by looking at things. And so you do the right thing, you say, I'm going to look at it. And so you can look at it in 2 ways. You could look at it in an electron microscope, just touch an EM grid to this, and let whatever is in that thing get attached, and go look in the electron microscope. Or you can shine X-rays on this and look at the X-ray diffraction pattern and see what you might learn from X-ray diffraction. So we've done both. Okay, so by electron microscopy, it was solved immediately. You look in the electron microscope, what's the gel made of? It's made of polymers. See it right in front of your face. So immediately, what was happening is we're getting a gel because this protein is making a polymer. Now that makes a lot of sense. For example, with actin can be in a soluble state or a polymeric state, and when actin polymerizes, it gels. So it makes a lot of sense as to why when these proteins form a polymer, they form a gel. Second thing is we can look at them by X-ray diffraction, and we see 2 diffraction rings, one at 4.6 to 4.7 angstroms, a second one at 10 angstroms. And between these 2, it told us exactly what it was. And what it was is a cross-beta polymer. And so these have been known since the 1950s, or 1940s even. Atzberry, and even before Atzberry, Linus Pauling predicted this, that proteins could interact such that an extended beta sheet of one polypeptide could interact with the next beta sheet via very predictable forces that I'll show you on the next slide. So this was exactly what we got. We got cross-beta polymers. We got diffraction rings at 4.6 to 4.7 angstroms that represent the distance between this protomer, the beta sheet of this protomer, and the next beta sheet, and the next beta sheet, and so on. So that's the 4.7 angstroms. In different cross-beta polymers, this distance is variable. It can be anywhere from 6 to 13 angstroms, depending on what cross-beta you're talking about. In our case, every time it's 10 angstroms. And we don't understand why it's always 10, because we've done this with many of these polymers now, and it's always 10. So the 10 angstrom observation is going to end up being important, but in ways that we're yet to understand. So here's what Pauling predicted. Here's a polypeptide chain right here. Here's a second polypeptide chain. And the main chain of the amino acid creates opportunities for hydrogen bonding between hydrogens and oxygens periodically along the chain. And so from first principles, Crick said this is going to be the way that proteins can interact. And he was exactly right. So that's all that's happening here, is that we're getting polypeptide chains to create this cross-beta interaction via hydrogen bonding. So maybe my biggest discovery before this, if this ends up being a discovery, was the discovery of something called the leucine zipper. And all the leucine zipper was an alpha helix. And so if that's your best discovery, that's pretty boring. So my second discovery is going to be cross beta, which is also really boring. But I think it's going to, I'm hoping it's going to be important. Okay, now the trouble starts. Now everyone says, Steve, you can't be right. This is impossible. And they said it's impossible because this sort of interaction is the pathogenicity of many forms of neurodegenerative disease. Prions, they do exactly this. So everyone knows about prions and amyloid fibers and tau and alpha-synuclein. And so all of that is bad news. That's terrible. You know, that's pathology. That's not biology. And so everyone said, Steve, you can't be right on this. And I said, no, I'm right. And I said, I'm right because of what's shown here. So if you take a pathogenic polymer and run it on a gel and expose it to SDS, even in the presence of 2% SDS, the pathogenic polymer won't come apart. It's stuck like to death. It can't ever come apart, and that's why it's bad. It's really bad. Morphologically, our fibers look just like these. This happens to be the sub-35 prion that Reed Wickner has studied so beautifully. So we look at, in the electron microscope, ours look just the same, but now we do this experiment, and ours come apart as soon as we dilute them. So morphologically, they look the same, but fundamentally, they're different. They're labile. They come right apart. So what we had to do, and this was really hard, we had to solve the structure of our fibers to explain why ours are that way and these are that way. People have already solved these structures, and they're long extended cross beta fibers, and so they understand the basis of this. But we had to solve ours to find out why ours are labile. And we've done this over the last 3 years in a wonderful collaboration with a physicist by the name of Robert Tico at the NIH, who literally invented solid-state NMR spectroscopy. And the only way you can solve a fiber like this is by that method. You can't do solution NMR. You can't do X-ray crystallography. The only way you can solve a fiber like this is solid-state NMR. And so we worked with him, and Rob is just phenomenal. So we can take our samples, and we get a beautiful spectrum. So we know we have a great structure. That's the good news. The bad news is, whenever we would try and assign peaks, let's say that's a glycine serine. And we knew it's a glycine serine, great, but there's 17 glycine serines in the low complexity sequence. So we don't know where to assign it, because there's 17 of them. You might assign it here, you might assign it here. And so it made it incredibly difficult to solve the structure, and the way we did it was a beautiful divide and conquer approach that was done by Masato Kato in my lab. And so the way he did it was as shown here. So we could label the whole protein with heavy nitrogen and carbon and get our spectrum. And it was too confusing, so we couldn't assign anything. So what we began to do is label half the protein and then fuse it with the back half unlabeled using entene chemistry. And this was predictable. You should be able to do this, but no one had ever done it before, because we had to get 20 milligram preparations, and Masato climbed the mountain and was able to do this. And so now we could have this half labeled, that half unlabeled, or reciprocally, the front half unlabeled, the second half labeled. And then once we began to find that the fiber core was in here, we could further subdivide and work our way towards the structure. So I'll just show you one example of that segmental labeling. So here's the whole protein with all its peaks. This protein has only the front half labeled, and all the peaks are in the front half, none are in the back half. So that would allow us to focus on the front half of the low complexity sequence to make the assignments, and by further reductionism, we can get the structure. So we've been able to get the structure, and it's fantastic. So here it is. And it gave the answer. And the answer is, yes, it's cross beta. We're right on that. In register, everything's right. But there's no continuous beta structure. The longest continuous beta structure is 3 amino acids. And every one of those little short ones is broken by non-beta. So you've concocted exactly the same structure, but you've built it to be labile. So you've built it to tip just right. So we further know, now we have many mutations that we know enhance the stability, and by having the structure, we can see exactly why that amino acid enhances the stability and causes ALS. We know 4 sites of phosphorylation of the protein kinase enzyme that takes this apart. It's called DNA PK. And those 4 serines, we know exactly where the serines are, so we know exactly how phosphorylation takes it apart. So by sticking with it for 3 years and working with Tico, we've been able to get what I think was the major shortcoming, and that's an understanding of why these structures are labile. And I'll just show you, by comparison, the structure of a pathogenic prion. This is a solid-state NMR structure of alpha-synuclein. When this guy polymerizes, it creates a prion that causes ALS, and the cross-beta structures are continuous for as long as 14 straight amino acids. So we've got the same structure, but ours, instead of having continuous cross-beta, has interruptions such that any particular cross-beta is no more than 3 or at most 4 amino acids in a row. So I'm going to end now. Hopefully I haven't gone too long. Okay, I'm going to end maybe early with some ideas. So here are the concluding ideas. These low complexity domains of DNA and RNA regulatory proteins function by forming cross-labile, cross-beta polymers. That is what we think allows these guys to form these structures. So the structures, and there are thousands of these. And so to really work out all the details, we now have kind of a roadmap, a way of thinking about it. So that's the way that we think that they work. What I'm predicting is from the birth of a transcript in the nucleus to its eventual translation, the genetic information may be sequentially passed through an informational cytoskeleton. So I don't think anything's floating. So from the gene to the nuclear speckle, movement out of the nuclear pore to the RNA granule, movement to wherever the translation, nothing's ever floating. It's always in this kind of informational cytoskeleton. So you're not messing around with your precious genetic information. You're passing it in a very discrete way through this process. 20% of the proteome is devoted to this task. Virtually all of our thousands of regulatory proteins have LC domains. So this is all over biology. This is not just one case here. This is all over the place. Low complexity domains are a cellular sink for post-translational modification. So cross-beta polymerization of these sequences in cells will be dynamic. So when people have done proteomics and asked what part of the proteome is post-translationally modified, 80% of post-translational modification is in this 20%. So this is where all the action is. This is where things are happening. And then the last thing I'll say is that mutations that enhance polymer stability can lead to neurodegenerative disease. And now that we have a structure of the low complexity domain of fuse, we can begin to understand in atomic detail how that works. So I want to close by saying I run a little lab UT Southwestern. I've always had a small lab, 4 or 5 people. And Masato Kato, Tina Han, Shan Shi, Ilman Kwan, I can name them on my hands. So 5 or 6 people have heroically done this. And we've been all alone in this stuff. Now, I think it's beginning to capture attention from other people, but it's due to these students, these trainees are the ones that did this. And I'm incredibly indebted to them. With that, I'll stop and answer questions. Yeah.
That was magical. 2 questions. One is the way this begins to describe sort of the fluid mechanics of a cell, you alluded to, but then sort of dropped away from, which is sort of looking at how these form and how it describes sort of the 3-dimensionality of a cell in a way that I hadn't seen explained before. So that was one thing, and you sort of alluded to it and then stepped away from it. And then the second thing was sort of a corollary to that. When I work in ALS, and the point that the stability is enhanced in all of or many of the proteins that are involved in neurodegeneration makes sense, but I'm curious how you think that relates to actually being causal in the mechanism, because I don't think it's as simple as that they're just hard to take apart. There's something else going on, and I'm curious if you think it relates back to that fluid mechanics part. That.
All right, let's deal with ALS first. I think the human genetics of ALS is going gangbusters. So they're finding these mutations that are causal. I mean, these are the mutations. And that is a gold mine for us because we have the assay. So we can simply ask the question for any of those mutations, do those enhance the stability in a manner that we could understand from the structure? So we are being fed by them, so it's terrific. So we've got structures coming for 3 or 4 of these that are going to allow us to broaden that out. And it's not just mutations in these low complexity domains. So we've worked on a form of ALS called C9orf72, and that's a repeat expansion that ends up getting translated aberrantly into dipeptides that are polydipeptides. And the polydipeptides, we showed those are the cause. And there are 2 of them that cause it, poly-GR, glycine arginine, or poly-PR, proline arginine. We published that in Science, and it was all based on this. This is how we put it together. Everyone said, oh, he's smoking dope. This guy's crazy. Well, now the whole field is doing it. They know that's the cause. And so what those do is they bind to these polymers. So PR can interact with an iterative polymeric substrate. So the weak interaction of each PR, if iteratively replaced, can allow for strong binding. And that, we think, does the same thing. So you have a polymer that's supposed to be labile, but if you bring on a PR-like protein that enhances the stability, you're doing really the same thing as you are as by making a mutation in the low complexity domain that creates the polymer. So I think we're laying the groundwork for people to understand how this really works. Now, what's to me incredible is those mutations are in the patient for 40 years, 50 years. Now, maybe certain forms of C9orf happen earlier, but most of the mutations that are changing these proteins, they're fine for 40 or 50 years. And then something with age, it begins to manifest. And people can see they begin to polymerize in aggregate. So I think we're laying the groundwork for the people who study the disease more carefully. We're laying a groundwork for at least a conceptual groundwork for them to think about. So that's the ALS part. Do you want me to try and answer the other part? Okay, so the other part is this idea of information transfer being solid state. That's very hypothetical. That's just my idea. I'm conceptually thinking that things are not left to chance. They're not floating in solutions. And this offers a way to think about that. it's so far in the future to really get down to the nitty-gritty of understanding that. But I'm throwing that out to be provocative, to get people to think that maybe cells are not just fluid and water floating, but that these sorts of things, these sort of low complexity sequences give an organizational, informational cytoskeleton. Yeah.
Can I follow up? Is it, you're next, whoever you are. So
Thanks, Larry.
So, on the one hand, so I want to make a small speech and then ask a question. It's a very small speech. So, your friend and mine, Mort Meyerson, reintroduced this, and then you sent me all these papers. And I'd like to confess that I feel like an asshole because this was the most exciting science that I didn't know about because I didn't read your damn papers.
It's okay, Larry.
And so I will never make that mistake again. I've been busy, but so the shocking part of this, I think, maybe, is that the place. First of all, I thought I knew the human proteome pretty well. I didn't know shit because this is 20% of it, meaning there's 5,000 proteins that have these things hanging on them somehow. If you do that, I'll punch you. So don't say that. So the shock is, what might be that you know where most of these sequences sit in biology, in human biology, and so it's great to think of them in these long neural things. I don't know many, but then Susan Strom's thing and Bill Wood's thing was not that. It was a different kind of migration. So it'd be good if you told us not about the disease where you stabilize them, but where these things are used in biology, because I think it's important.
Yeah. Okay, so Larry, I mean, I've always been focused on gene regulation. I've always been interested in transcription factors, and maybe I'll go to translation, but only if there's nothing else to do. Very transcription-centric. These are all over the cell. It's not just this. So now we know every intermediate filament protein has a coil and has a low complexity domain on the front, a low complexity domain on the end. You can make your coil, but you can't make the intermediate filament without these. So this is at the heart of how you make intermediate filaments. Intermediate filaments are what the RNA granules bind to. That's known. So this is all over the cell. And you know, it was a problem in cell biology that you just couldn't work with because you can't get function without form. So if a protein's misfolded and floating, you can't get home. And so it might have been that these proteins, and people still argue that these proteins never fold, never do anything. But if that's true, good luck. But once you can say, here's what they do, now you have a way of beginning to dissect it. And so what I want to emphasize is this is not just in transcription. This is all over cell biology.
So that was beautiful work. So the example you showed us was a homopolymer. What's known about heteropolymers? And I'm over here, Steve.
Oh, okay. Oh, yeah.
What's known about heteropolymers, and in particular the specificity? I mean, if 20% of these proteins have these domains, I'm sure they don't all interact with each other.
Oh, no. Yeah, but you laid the table for me, and I gave myself a little extra time. So that's a really good question. So what we do is we make a little fiber and we sonicate it. Like M-cherry fuse, we have a seed. And then we add some GFP fuse and let it grow. And then we put it in the turf microscope and say what happened. So the little M-cherry seed grows green from either end. So that polymer can grow on itself, okay? Now we do the exact same experiment with the closest paralog of fuse, which is Ewing sarcoma. It has the same structure. It gets translocated in cancer. It's all the same thing. And you do the same experiment, and it will not extend. So there is very, very strong specificity in what you make. Now what happens is instead of elongating from the end of the seed allows the EWS to form a polymer right next to it, its own polymer. So we are on, at least we think we're on the primary level of understanding, and that is the initial cross beta. There's going to be secondary levels. There's going to be much more to this that we don't understand. So when we look at fuse, only part of it is in the fiber. The fused low complexity sequences has loops coming out that are not part of making this fiber, but they're probably important for that next level. And we don't understand that yet. But it's going to be a really fun decade or two for people to get all the rules and regulations, because if we're on the right track, people are going to be able to figure these things out.
Taking the bait on the information cytoskeleton, where do exosomes fit into this?
You know, this is an embarrassment, but I don't know what exosomes are.
Vesicles that package various substances, including RNA sometimes. They're found in blood, and they're not well understood, but there is the notion that there may be some information transferred that way.
That I don't even know about them. So maybe you can tell me about them this evening. Yeah.
So I know nothing about this topic, but the one thing that strikes me, which is kind of interesting, and I'm probably, particularly in this audience, using not the right words, is adaptability. Because what you were describing was a structure that inherently breaks down, and presumably in a context it breaks down. So it's temporal, but structures are also being formed, so it's adaptable, whereas with the prion, the structure becomes fixed, and the adaptability is lost. Is that a fair statement, that this may be a dynamic?
No, you're exactly right.
Okay.
If our fibers were permanent, I can't see how you get biology out of permanence.
But this goes back to all the other things that we're learning about, which is epigenetics, the role of microbes, and that the body is so dynamic and potentially adaptable.
Yep.
And this is probably part of that, that dynamic, it would seem.
No, I was talking with Tom before the session started. This is the most primitive way proteins can work. So the very first proteins in evolution only had one amino acid, because you didn't make two amino acids before you had one. So the first protein ever made was a homopolymer. And that homopolymer did this. That's all it could do. It could do, because it had a peptide backbone, it had all the opportunity for hydrogen bonding, just as Pauling told us. So that first protein, that's all it did. And maybe it phase separated something. And so here we are, after 1 billion years of evolution, we have these terrific, well-folded ribosomes and RNA polymerase, just unbelievable. And what do they have stuck on them? This junk, this just pathetic junk. And yet the pathetic junk, I think, is a remnant of how proteins were first used. And it's just barely enough to help a little bit, organizationally a little bit. Phase separating.
So I'm sorry. So one of the challenge then is to try to understand the context in which these changes, that dynamic occurs.
Yep.
And that's going to be okay.
Yep.
Yeah. Thank you very much.
Yeah, thank you. I think we're out of time, and you can find Dr. McKnight thereafter. Thanks.
All right. Here.